Welcome back to our exciting journey through the bacterial cell, and recall that when we left Norbert last time, we had just talked about the nuances of the cytoplasmic membrane. Today, we're going to zoom into the cytosol, and we're going to take note of some of the structures that exist here. Recall this sort of glob-looking structure, the white one that you see there. That is called an inclusion, or occasionally an inclusion body, and they go by many names, but their primary function lies in regulating osmotic balance, in storage of macromolecules that a cell might want to hold on to. You can think about the types of those. And additionally, they often play a role in buoyancy. We'll talk about those inclusions that allow a cell to float or sink to a desired level if it lives in an aquatic environment. Let's begin with those that play a role in storage, and I want to make note that sometimes these are actually bound. Now, they're not bound by a true phospholipid bilayer, and in fact, that is one of the things that differentiates a bacterial cell from a eukaryotic cell. Bacterial cells do not have true membrane-bound organelles, but sometimes these storage granules are bound by a single layer of phospholipids. So rather than a true bilayer, we might just see this single layer of phospholipids, a monolayer, and sometimes interspersed within that monolayer, we might see proteins. But do take note that that's a single monolayer and not a bilayer. These are usually about 2 to 4 nanometers thick, and they will bound these storage inclusions. Not all inclusions are bound. Some of them are unbound. Let's look at some of the types. We talked about glycogen storage granules. Glycogen, of course, being the sugar polymer, right? Many sugars all bound to one another, forming this glycogen, large glycogen polymer used for carbon storage, energy storage. Polybeta hydroxybutyrate is not unlike glycogen. It's very much the same. It's a carbon polymer that's a good way to store carbon. We also see granules that store polyphosphate. Polyphosphate is a polymer, a long polymer of phosphates, and it looks something like this. So our chemistry nerds are going to dork out on this structure, and you can see basically you've got multiple phosphate groups linked to one another. So notice that there's some really high energy bonds in anytime we see multiple phosphates linked to one another. These are actually called phosphoanhydride bonds right here. So notice that these bonds are very high in energy, very energetic, and there's another one there. And so we can see a bacterium wanting to store long polymers of phosphate because it is a very energetic storage molecule. It also is an important storage molecule because so many things in a cell are made using phosphate groups. Think about ATP, which is adenosine triphosphate. It is made using a phosphorylation of ADP in order to make that high energy, universal energy currency in the cell. The next type of storage granule is a cyanophysin granule. This actually stores amino acids, but the reason that it stores these 2 amino acids, aspartate and arginine, is because they're high in nitrogen. And so this is a good way for the cell to store nitrogen. So another reason that we might see these inclusions, another type of inclusion, shall we say, is a microcompartment. It was a long time before microcompartments were studied and understood. In fact, this wasn't in. If you had taken my class even 7 years ago, you would see that this wouldn't have been in the notes. Microcompartments are argued by some scientists to have some analogous function to an organelle. But what's very different about them is that they are made up of protein. That is, they're encased by a protein polyhedron. And so they're able to form gradients across, but they're not membrane bound. They're just protein. So you can kind of envision a very regular repeating structure, a polyhedron. And in there you will see that there's not only storage of certain molecules, but also some sort of functional role that these microcompartments play. For example, in cyanobacteria, we see carboxysomes, and carboxysomes are actually the site of carbon fixation. That literally means taking inorganic CO2 and using that inorganic CO2 to build sugar, right? They're very building or biosynthetic process that takes place in these carboxysomes. So that's some pretty cool chemistry as well going on there. As we explore this next inclusion, I'm thinking about both Joanna and Katie, who expressed some type of interest in the ways in which changes in the earth affect bacterial life. And I think you'll be very interested to know that, in fact, the earth also is something to which bacteria can sometimes respond. That is, there are bacteria that are literally magnetotactic, meaning they can orient with respect to the Earth's magnetic field. They can be either north-seeking or south-seeking, and that means they have inclusions that are packed full of metal compounds: iron sulfide, fool's gold, greigite, magnetite, so iron oxides, for example. And this is a cool picture of some of those magnetosomes. This is found on University of Alaska Fairbanks website at .edu, and it shows you some of the regularly repeating structures of those magnetosomes and how these bacteria then that contain these magnetosomes can be south-seeking or north-seeking and really utilize the Earth's magnetic field in order to orient. Now, there is also on this page an incredibly cool video, and I'm going to go ahead and play it. These are photomagnetotactic bacteria, meaning that they orient both with respect to light and with respect to the Earth's magnetic field. So check this out. Holy cow. Absolutely incredible to think about bacteria who are being that responsive to that basic feature of the Earth. As I think about this next inclusion, I always think about balloons, and thankfully I have some nice ones here to demo it. Gas vacuoles store gases, and as you might guess, the way that a balloon would float in a swimming pool, these allow for buoyancy. And in fact, they're actually comprised of proteins. So picture long helical proteins encasing gas, and that is what this balloon would be analogous to. So as you might guess, the more gas vesicles there are, the more a cell will float, and the higher the level within the aquatic environment. And of course, if a cell wants to float to a particular wavelength of light, it can either construct or deconstruct these gas vesicles in order to accommodate that. Now, it's really interesting to note that these gas vesicles, so if you took the red balloon and the blue balloon, each one of these is an individual gas vesicle. But if we put them all together with the pink balloons and we have this conglomerate, this whole thing together is called a gas vacuole. So gas vacuoles are aggregates of large numbers of gas vesicles. Now, as you might also expect, if you take a bacterium, an aquatic bacterium, perhaps a cyanobacterium that's filled with these gas vesicles, and you put it into a high pressure environment, for example, you put it into a bottle which you put a rubber stopper on and you hit it with a mallet, well, what's going to happen to these balloons, or AKA the gas vesicles? You got it. They are going to totally pop. And that means that what we can actually do is to control bacterial populations with something called sonication. And I thought this would interest a lot of you. This actually is a pretty cool website for the Amy H. Remley Foundation looking to restore the waters. And I think what's really neat about it is that they show a lot of those demos that I just mentioned regarding gas vesicles. Additionally, they show some really cool pictures of gas vesicles, but I think it's also really neat that they talk about the collapse of these under high pressure and the fact that they found ways to use ultrasound to control the growth of cyanobacteria, which are unfortunately mislabeled algae. They're not algae, they're bacteria. But it is cool to look at some of those pictures, and in fact this one here I think does a great job. You can really see the conglomerates or aggregates of gas vesicles that do look like if we were looking at those balloons end on in a cross section, and how they conglomerate together to form a gas vacuole. Maybe this is one of my favorite of all of the inclusions, and certainly one that is extremely important in supporting a cell's livelihood in going to the right level within an aquatic environment. As we end our coverage of inclusions, I just want to take a moment to begin to entertain a question that you can continue to think about. That is, what will affect the number of inclusion bodies seen in a cell at any given time? Now, broadly, I think we could answer this question with environment. We know that the environment that surrounds a cell is going to dictate what it can either store away or what it has to deplete. So we could say the level of nutrients in the environment, so concentration of nutrients. As you might imagine, if a cell is in a very nutrient-rich environment, it can store away a lot of the food as, say, glycogen in a glycogen storage granule. It can save it for a time of need. We also understand that maybe the wavelength of light or the type of energy that it is gaining in that environment will affect the number of inclusions. So with regards to our gas vacuoles, we know that the wavelength of light will be what dictates the depth that it takes within an aquatic environment. So maybe the wavelength of light or light penetration into the aquatic environment certainly is going to make a big difference. So at any given moment, it's all about what a cell is surrounded by, which will dictate, just like with us, will dictate what is going on internally as well. You can continue to think about this question and maybe ponder it more deeply. So let's return back to Norbert's cytosol and further take a journey here into some of the functional components within the cytosol. You'll notice that there are these little blue dots, and in our model, these little blue dots are supposed to represent the ribosomes. The ribosomes are, in my mind, one of the most highly misunderstood cytosolic components. They are the factories of proteins. That is, they make proteins, but often a ribosome is typified as simply being the workbench upon which proteins are constructed. Nothing could be further from the truth, because in fact the ribosome itself is highly active in the process. In fact, as it turns out, the RNAs in one of the RNAs in the larger subunit of the ribosome is actually what makes the bonds between the amino acids in the polypeptide chain that is going to become a protein. So these are incredibly highly dynamic in the process of protein synthesis. So that gives you some idea of what a ribosome is made up of, right? It has to be made up of RNA, because we know that one of those RNAs acts like an enzyme, a ribozyme, that actually synthesizes the bonds between the amino acids as it constructs those proteins. So we know that one of the things that it's made up of is RNA, the other thing is made up of is protein. And the ribosome as a whole has a small subunit. Here in my cheesy picture, we see a 30S ribosomal subunit. Here is a 50S ribosomal subunit. Together, they come together to make a 70S fully functional ribosome. And the 30S small subunit is composed of both RNAs and proteins, and the 50S subunit is composed of both RNA and proteins. So there are two macromolecules that construct a ribosome overall, RNA and protein. So looking at the maybe confusing part here, and I have a feeling that some of you are thinking, wait a minute, Rachel, either I'm like really hungover or 50 plus 30 doesn't equal 70. But the reason for this is that the units that they are measured with are units called Svedberg units, and Svedberg units are based not only on molecular weight but also on volume and shape. That is to say, how aerodynamic the molecules are. So what we're measuring is if we take a ribosome or a ribosomal subunit and we put it in a super fast centrifuge called an ultracentrifuge, we're actually measuring how rapidly does the ribosome sediment down to the bottom of that ultracentrifuge that's spinning really, really fast. And we know that that's going to depend not only upon the molecular weight of the molecule, but it's also going to depend on how aerodynamic it is. So its volume and its shape is also going to matter. That's why these are non-additive, is because it doesn't just depend upon the overall molecular weight, it also depends upon those other qualities of the molecule. So we can note here in this kind of cheesy picture that both bacteria and archaea are typified by a 70S ribosome. They both have a 30 and a 50S subunit, which is very different from eukaryotes. Eukaryotes have a different sized ribosome. They have an 80S ribosome rather than a 70S ribosome, but archaea and bacteria both have a 70S ribosome. However, bacteria and archaea differ just a little bit. For example, some of the types of RNAs that you see in archaea are different than bacteria. For example, we see a 5S rRNA in bacteria versus a 5.8S in archaea, which is actually more like eukaryotes. We see a different number of proteins. In fact, archaea have more proteins in their subunits than do bacteria. So despite the fact that they both have 70S ribosomes, we see that they do differ just a little bit. However, maybe arguably the most applied and important difference to remember is that between eukaryotes and prokaryotes. And that is simply because, think about it for a minute, what can we do if we know that bacteria have a different kind of ribosome than do eukaryotes? So said another way, a bacterial infection that might be inside of me has a different ribosome than my ribosomes. What application comes really clearly from that? Why that is such an important and applied difference? Why is it something that we constantly utilize in the medical pharmaceutical industry?